think will be a very fascinating discussion. My name is Greg Hillier. I'm the editor-in-chief of Progressive Farmer Magazine. I will be your host and also will be fielding questions from the audience. Today's webinar is Digging into Soil Health, the Role of Soil Microbes. And you know, researchers and growers have been learning that what happens below the soil surface is really just as important as what happens above it. And part of that equation is soil health. And there's many factors that affect soil health. And one of those is soil microbes. But I know, like many of you, you probably have questions on just what these soil microbes are. You know, how do they benefit crop performance and yield? How can they help suppress disease and maybe help crops be more resilient? So really, to me, it's kind of like this new frontier in production agriculture, and we're just now getting our hands around soil microbes and what they can and can't do, and kind of unlocking the secrets and mysteries of soil microbes. So our speaker today is going to help us understand the role of soil microbes and help us separate that fact from fiction. Our speaker is Dr. David Stark. He's the President of Agriculture for Holganix. It's a company working in the soil health and soil microbe arena. David has a PhD in molecular biology, and before he came to Holganix, he held various positions at Monsanto. So following David's remarks, we'll take questions from the audience. On your screen, if you look at the bottom right-hand corner, you'll see a little speech icon. If you click onto that, you can type in a question, and you can do your questions at any time during the presentation. And once David has finished his remarks, then we'll open up to your questions. So with that, I'll turn it over to David.
Okay, thanks, Greg. Thanks everyone for attending. So like Greg said, I'm Dave Stark. And I'm going to start a little bit differently maybe than from a lot of speakers. I'm going to start with an apology and a confession. So my apology is, as a scientist, I want to help you understand all the different things going on in the soil and the products that people are selling you. It's not my intent to sell. My apology is the best data and pictures I have are with people that have used our product or paid research with it. So it will be an example of what a product like ours can do. But again, I hope you see it. It makes the point and reinforces what I'm saying about different types of soil microbes they can and can't do. And my admission is, you know, as a scientist and as Greg said, an executive at Monsanto for my whole first career, I did not think very highly of soil microbes. And I started as a favor to a friend consulting this company called Holganix, which was a turf company and wanted to get into agriculture around 2015. And in my opinion at the time was microbes are bugs in a jug, and there's other less polite terms that I've heard, but I did it anyhow. I can happily say in that time, I've not only learned a lot, but people are finally starting to do really good research on soil biology. It was an area that probably wasn't well funded, but a lot has changed in the few years to the point where, you need the next page, buddy. I thought we did this before. There we go. There's a lot we could talk about. We're not, don't worry. But these are all things that the soil microbiome does. You know, for example, there's some really good science that says the soil biology contributes 7 times more to protecting a plant against insect damage than the plant's own genetics. I mean, isn't that wild? But what we're going to talk about is nutrient uptake and cycling. And that's a big part of what microbes do. And we'll just stay focused on that. This other stuff I'd love to talk about, but maybe in a different webinar. So why nutrient cycling? Nutrient use efficiency, as you all probably know and have heard, is pretty poor. This isn't to say on your ground, in your operation, this is what's happening. These are averages across the country. But it's pretty safe to say less than half the fertilizer we apply goes to the crop. Nitrogen use efficiency, it says 40 to 65%. Candidly, that's pretty generous on some ground, and some ground actually does better. Phosphorus is terrible because it's quickly reacting with other things in the soil and converting to a chemical form the plant can't access. Same with potassium. Potassium also can move with water. So when we consider that in 2021, the USDA calculated that 36% of the operating cost of growing a corn crop is fertilizer, and that it's about a $20 billion market, and this is not current, this is a year ago, this means we're wasting more than $10 billion on fertilizer that's not doing what we want. So I think this is a pretty important topic for us. So I'm going to talk about nitrogen and then phosphorus and potassium. So if we look at nitrogen, there are products out there that try to add nitrogen to the soil as one approach to the nitrogen issue through microbes that can take it from the atmosphere and convert it into ammonia. And then ammonia can be used to make proteins, etc. So that's the key. 78% of the air we breathe is nitrogen. Why is it so hard to use? Well, if you look at the top, you see the two ends and then the three lines joining them. That's showing that the nitrogen gas is two nitrogen atoms, triple chemical bonded. If you go to the bottom and you see the N with the H's around it with a single line, those are single chemical bonds. The triple bond in nitrogen is one of the strongest triple bonds on the planet. It is extremely difficult to break. And to make nitrogen gas into ammonia, we have to break that triple bond, which takes energy. What kind of energy? Well, lightning. So in a thunderstorm, you actually get a little free ammonia and nitrate. Lightning is 50,000 degrees Fahrenheit. So there's a little bit of energy in that. We have a process called the Haber-Bosch process, where we take nitrogen gas. Now, we can't build a vessel, I don't think, that can be 50,000 degrees Fahrenheit. That's much, much hotter than the surface of the sun. Instead, we take nitrogen gas, and we're only putting it at 750 to 900 degrees Fahrenheit, but 150 to 250 atmospheres of pressure. That's a lot of energy, and that can bust apart that triple bond and make ammonia. So when we ask microbes to do this, microbes have an enzyme called nitrogenase that can do it, and it does it well, but it requires energy. So that's probably the biggest challenge in this. So when you put a rhizobia inocula on your soybeans or other legumes, that is pretty efficient at taking nitrogen from the air and making ammonia, because the plant makes a nodule. It pumps a lot of energy into it, which the bacteria need, so the plant takes care of the energy needs. And nitrogenase as an enzyme is poisoned by oxygen. So the plant removes the oxygen so the bacteria don't have to spend the energy doing that. If you cut open a nodule, what color is it? It's pink or red. That's called leg hemoglobin. It's similar to the hemoglobin in our blood. What does hemoglobin do for us? It binds oxygen, carries it through the body. So you can see a good active nodule has that hemoglobin type molecule because it's binding oxygen to protect this process. Now, there are free living bacteria that don't live in a nodule, that can live in the soil, and there are companies that are selling those that also try to take the nitrogen from the air and make ammonia, and they can do it. And I can tell you, there are a lot of bacteria in the soil that will do this process. If nitrogen is present, they won't. It's just biochemical feedback, because why spend this tremendous amount of chemical energy to make ammonia if it's already there? I mean, it's just common sense, and that's how biochemistry tends to work. So there are some products where they've genetically changed the bacteria, so even if nitrogen is present, it's still trying to take the nitrogen from the air. And I think that's clever as a molecular biologist. But this quote in the box in the lower right sums it up, that those bacteria are spending a lot of energy to pump oxygen out and break that nitrogen triple bond. that makes them far less competitive with other microbes in the soil because they're all competing for the same food. You know, I make the analogy, it's like two really good marathon runners and you put them at the start line, but you put a 70-pound backpack on one. You know, who's going to win? It's pretty clear. So this is one way of doing it. There's a lot of technical problems. You know, just to help you appreciate, you know, sadly, ammonia fertilizer, what happens if it goes back to nitrogen gas? It's a bomb. So because that energy then gets released. So I want you to appreciate this is a very energy intensive process. Another way of helping nitrogen in the soil and nitrogen use efficiency has a lot of science around it that's pretty recent. So this is a study that actually is a meta study, meaning it looked at 230 peer-reviewed, published scientific research looking at nitrogen use in corn, rice, and small grains. So this is a lot of science. In a biologically inactive soil, we've got what you see on the left around the black box where we put fertilizer on, the crop takes some up directly, and a lot is lost. It's, you know, to the environment, it runs off into the water, whatever. But when we have biologically active soil, what really happens is the microbes, which are more plentiful and aggressive than the crop, actually eat that nitrogen first. And that's a good thing because soil biology tends to live within a half an inch of a root. So they immobilize it in the root zone. And as long as we have diverse biology, which we're going to talk about, we're going to cycle that nitrogen back to the crop. So we can really increase the efficiency of our nitrogen to where the conclusion is biologically healthy soils produce greater corn yields per unit of fertilizer input. So when you use a microbial product to help you retain nitrogen that you're either buying and putting down or you already have in the ground, you want to have less nitrogen overall because there's another point. Plants won't feed and attract microbes if they already sense excess nutrients, especially nitrogen and phosphorus. So you're going to see, you know, me talk a lot about if you want to get the most out of a microbial product, back off your nutrients because make the plant want to feed the microbes. It'll pay off in the end for sure. So exudates are increased in nutrient limited soil. Same story for phosphorus and potassium. So you probably have heard people talk about how there's a lot of P and K in your ground, not only in the top 6 inches, but probably down in the subsoil through years of applying. It gets tied up, but it's not in a form the plant root can take up. Microbes can. Microbes secrete different organic acids and chelators that can grab that total pool and unlock it. Many bacteria and fungi can do this. As a matter of fact, if you look at a biologically healthy soil, the phosphorus that's in the microbes is greater than what's in your crop. And again, that's a good thing because they're tapping into that soil bank. How long can you do that depends on your soil tests, but you can definitely dial back your P and K when you use a good microbial product. And there are products out there specifically designed to unlock phosphorus and potassium, and that's credible because microbes are good at doing that. Now, there are other products that will induce roots. And I've had a lot of people tell me, oh, you know, I saw more roots, more root hairs. I just didn't see anything on the yield monitor. But I'm here to tell you when it comes to nutrient use efficiency, roots really matter for a number of reasons. If you think of the soil within a typical root zone, the roots only explore about 1%. That's pretty poor. Most nutrients need to be in close contact with the root to be absorbed. The more roots, the more root hairs, the more soil explored, the more nutrients we take up. There are also fungi like mycorrhizal fungi, which are really good at getting in between soil particles your root hairs can't access. So they increase the effective surface area of your roots anywhere from 100 to 1,000-fold. So again, biology really helps you explore more soil and unlock the nutrients you've already spent money on in the past It's just sitting in your ground and not accessible. Another thing about roots, you see, on the right, look at all those roots that are breaking up compaction. When they die, they're making channels for air and water. Everything you want living in your soil needs oxygen and water. And this is going to help aerate that soil. It's also secreting exudates, glue to give you more soil structure. So again, if we look at building stable organic matter, which is so important on so many levels, and you all know that, roots contribute a lot more to that than the residue we leave on top of the ground. So roots are really important if you want to be on that soil health journey, which is going to help you be more efficient and actually make your land more valuable in the process. Well, so real quick, to summarize, microbes eat fertilizer, which is not a bad thing as long as we have enough of the different kinds of microbes, because they live in the root zone and they'll cycle it back to the crop. Microbes are absolutely, totally 100% required to unlock nutrients in your crop residue, manure, any other organic matter that's out there. And through time, working with roots, that's how we actually build soil instead of breaking it down. So I keep talking about microbes. I want to show you pictures and talk about what they are. Here's a list, and they go from the smallest, the most abundant, the fastest to grow bacteria. Some bacteria in ideal growth conditions can double every 20 minutes. They are the most voracious. They will get the first nutrient bite available in your soil. And then as we go down the list, we go a little bit larger, a little bit slower to grow: fungi, algae, protozoa, amoeba, nematodes. And yes, there are a lot of nematodes that do not eat your plant roots. They eat other things in the soil, and they're actually beneficial. All of these are required to build soil health. So this is not a component. You need all of these pieces. So now this is my second confession. Molecular biologists have a lot of expertise in evolution because DNA is one of our expertises. There are a lot of people out there that just will sit back and tell you in amazement, isn't it wonderful how through millions and millions of years of coevolution, all these things work? And I would have told you the same thing 20 years ago, even though frankly, I've always believed in God. I've always been a Christian. I just figured if God used evolution, who am I to tell him he's wrong? But what I've learned by following the science instead of what I'm being told is the data don't support millions of years of evolution. And I can go into great detail. The data actually supports Genesis chapter 1. And it's amazing that through this time, God was probably laughing at me and then revealed the intricate, elegant complexity that frankly mathematicians calculated years ago as being absurdly impossible to be random. And I could go into great detail. So I bring this up because I think it's really important that you hear from scientists that things are not what we're always taught to believe. A couple of weeks ago, I did this at a fairly large national conference, and a gentleman very respectfully came up and said, I hear your passion and the science leading you to say no to evolution, but I bet if you applied science to your faith, it would also disprove it. And I was disappointed because it means I didn't communicate clearly. You see, following the data, I'm concluding the Bible is right. I always had the faith. I'm actually following the data. And I would love to talk about this if you've got more questions. As I go through an example using nitrogen of what each of these microbes does, look at the complexity and how all the pieces fit together. It's a great example. So bacteria and actinomycetes. The picture there, kind of the greenish background, those are bacteria. Again, they're tiny. They come in different shapes and sizes. That's more of a close-up of actinomycetes in the lower corner there. They're also very tiny. They're very closely related. They do great things in the soil. They're necessary. They degrade organic matter. They grab your nutrients. They'll convert ammonia to nitrate, which is better in terms of plant uptake. They dominate tilled ground. And most microbial products you're being sold only contain bacteria, and they contain a few species. That's not necessarily bad, but as you'll see, it's only one piece of the equation. How can you tell? To me, it's really easy. If it's in a powder, or if it's a liquid in a jug that's tightly sealed that you could put on the shelf for months, it is not diverse biology. It's probably just a narrow slice of bacteria and some fungi. Now, here's a concept, bear with me, because it's all going to fit together. Carbon to nitrogen ratio. In bacteria, for every nitrogen atom, on average, there's about 5 carbons. As a biologist, what does that mean? That means these are nitrogen pigs. They tie it up. That is an incredibly high nitrogen requirement for these microbes. So let's move on. Fungi. So you can see here in the dirt, these fungal mycelia that go all through there. They're real tiny, like I said earlier. They can find water and nutrients between soil particles the plant root can't. They are better at degrading organic material, especially the woody stuff, like your corn stalks and wheat straw, and I'll show you why later. They eat bacteria. Look at all that white stuff, that's carbon, so they actually add carbon to the soil. Their carbon to nitrogen ratio is not 1 nitrogen to 5 carbons, it's 1 to 15. So about 3 times less. There's a reason. And again, some microbial products will contain fungi. Now be aware, if it says unicellular fungus, that's yeast. Yeast are not bad for soil. They're great for beer, wine, and bread, but yeast are not producing these hyphae. Yeast do not break down your woody residue like corn stalks. They're not doing all the things here that I'm talking about for fungi. And last, well, I'll talk about protozoa, amoeba, and even the beneficial nematodes, which I need to add a picture of. They're predators. They eat each other. They eat bacteria, fungi. A single amoeba can eat 10,000 bacteria a day. They all require oxygen, just like the fungi. There are a lot of bacteria that don't require oxygen, but fungi, except yeast, and the protozoa and amoeba and nematodes need oxygen. Their carbon to nitrogen ratio is now 30 to 1, 6 times lower than bacteria. They eat 10,000 bacteria a day. What's going on? Well, remember, bacteria are eating your nutrients first. They live within ½ inch of a root. As long as and they'll grow quickly, but as long as we have the protozoa and amoeba, they're predators, they're eating a lot of bacteria, but that nitrogen is toxic to them. They have to get rid of it. So they spit it out as ammonia and nitrate. Where? Within ½ inch of a root. The cycle is complete. So we need all these microbial types, and almost nothing, by the way, you'll buy in terms of microbial products will have protozoa and amoeba. Really, it takes something more like a compost tea type product before you get that. So again, the cycle is complete when we've got all this biology. So I get asked, once I've got really good soil, do I need to keep adding your product? And the answer is generally yes. There are things that happen that disrupt the biome. And frankly, if you were to monitor it, even in the same spot in your field, it's changing day to day, week to week. It's influenced by what you grow. So things like tilling, knock back fungi and explode bacteria, overuse of fertilizers, again, tells the plant, don't feed microbes. Pesticides, especially soil fumigants, kills everything. But there are things we have no control over. Freezing, because these things are all full of water. When ice crystals form, they expand, and that kills a lot of the microbes. Flooding. 48 hours underwater, the soil is depleted of oxygen, so the good microbes tend to die. You can tell when the flood water drains if you smell it before you see it, because it smells like the bottom of a swamp. But it's even more complex than that. It's not just the microbes that corn versus soy versus wheat attract. It's actually been shown corn hybrid to corn hybrid. The microbes in the root zone can differ by 6 to 10%. Same between soy variety and soy variety. So the plant is actively farming microbes, attracting what they want, repelling the others. So we have to have diversity to make sure the plant has what it needs. So a product like ours is really an engineered compost tea. It's not compost tea. It's similar to the process where we have a starting inoculum that has over 800 species of bacteria, fungi, protozoa, and amoeba. We DNA fingerprint, we can tell you every one. And in our process, because we change the conditions throughout, we can enrich for each type or family of microbe. So for this particular product, we enrich for those that are really good at cycling nutrients and secreting hormones to induce rooting and germination. It's a complete product. There's nothing else you need to add. It has sugar, a pound of molasses, food stuff for these microbes because they're alive. They're not a powder. Most of these will not form spores. It is actually a liquid that we stabilize through refrigeration. So your distributors have refrigeration. That is the best way, and frankly, the only way I know of to stabilize diverse biology. So if you're getting something that's not refrigerated or something that you don't inoculate and grow yourself on your farm, it can't have all this biology in it. It doesn't mean it's a bad product. It's just going to be more narrow in what it can do. So, we've got data on this. So this is these are turf experiments. This is a summary of 26 different studies done at North Carolina State, Penn State, and Purdue, different soil types, different forms of fertilizer, fast release, slow release, and we're looking at turf performance. And you can see, in the extreme case, 88% nitrogen reduction to maintain turf performance. It's not because the turf needs less nitrogen. It's 100% because the nitrogen that's been applied is staying in the root zone and a lot more is going to the turf. Now I have to, stress, don't use a product like ours and cut your nitrogen 88%. We don't remove nutrients because we don't harvest our turf. You remove nutrients every time you harvest a crop. We've got to put some back. My point is, this is a tool that helps you dial back your inputs to find that sweet spot between how much you spend on inputs and what you get in yield. And I'll show you some more data on that. So this is a pasture. This is in Alabama. This is the same field. It was all reseeded with fescue. It was all watered. But on part of the field, he added our product with the diverse biology. And that would be the bottom two pictures. And just look at the difference. What this says is a couple things. This field is generally poor in microbial activity because when we add microbes, we're releasing a lot of nutrients. We're also stimulating germination and root development and growth. So this is a nice visual sign that we need biological activity here. Now I've got a couple different studies. This is a corn one. This is 2 years in a row. This is the same field close to Champaign-Urbana, Illinois. This is a third-party, agronomy researcher. In the first year, he's rotating off of soybeans, and he calculated to get his 230 bushel an acre corn target. He only needed 45 pounds of UAN per acre, really low. And so we did an experiment where we cut it back 20%. Now that's only 11 pounds. What we saw is the same yield with 20% less fertilizer, but 11 bushels more when we put in the 45 pounds. Remember, this is all a really low rate. So we're not drowning the plant in nitrogen. The plant is working with the microbes to get that nitrogen that's in the ground. In this case, clearly it would pencil out, spend the 11, the money on the 11 extra pounds. Now the following year, corn on corn, now he's going more like 180 pounds of nitrogen per acre, 20% less. We got 9 bushels more. There it penciled out better to cut fertilizer because we saved on both ends. Less input, higher yield. Again, a tool, not a formula, but a tool. Same kind of deal here. This is a third-party trial in Iowa this last summer, and you can see the yield where we did 5, 10, and 20% less fertilizer versus check. We got about a bushel more with 20% less fertilizer. Here it may pencil out better to do 5% less because we got 6 bushels more. Again, it's a tool to help you really dial in efficiency better. This is corn stalks, and this is a grower in Indiana. Now we're going to see in a minute how potassium is a really important nutrient for stalk and stem strength. And so here toward the end of the year, you can see how the stalk is starting to shrink and it's senescing and pulling away. But where we have the biology that can unlock potassium in the soil, the stalk is much stronger. So again, we're seeing the visual signs that we're getting more of the nutrients into the plant. And of course, he got rewarded with better yield in this case too. I talk about germination and establishment. This is also from the summer. This is just from a customer. This is not a third-party researcher. You know, look at the difference, which is why if you're going to use a product that is, and there are a lot of microbials out there that will induce germination and rooting, put them down when you plant or as close to planting as practical. Don't wait till V3 or V5 because we miss this. In this case, that supposedly is the best check, that most of the checks hadn't emerged, and that's the average treated. You should see this because remember, microbes live in the root zone. It's their home. Plants feed microbes. They'll spend up to 30% of their energy attracting and feeding microbes. So microbes are a lot like people. They want a bigger home. So they secrete hormones to induce the rooting. And you know, the quicker you get that out of the ground and that root mass underneath, the better that's going to withstand whatever stress comes at you. This is a really important picture for a number of reasons. I like it because it's something anybody that owns a shovel can do, and I bet you've got a shovel. It's free, and it tells a lot about your soil. So here we're comparing on the left three, where we mix our diverse biology with Pivot Bio. That is a microbial that is intended to take nitrogen from the air and add it to the ground in the plant. Plus we had nitrogen fertilizer. The middle two are the Pivot Bio only, and the right is the nitrogen only. So when I look at this picture, there are a couple things that jump out. First of all, look at the nitrogen. The roots are pretty bare. There's not many root hairs. You probably can't see it, but I've really zoomed in on this, and the grower even said, look, that where he's got all three products together, the plant's taller, greener, the roots are bigger, and a lot more root hairs versus the other two. This tells me this soil is not biologically diverse because root hairs and that dirt that sticks to the root requires diverse biology. It's a process that's called quorum sensing. And basically what happens, my soil microbes are always sending out and receiving biochemical signals. They need to get signals not only from enough individuals, but enough different kinds that once they hit that threshold in quorum sensing, they change their gene expression. And what they do is they start working with the plant root exudates to start gluing soil particles together. And that is what you see on the left. That dirt on the root is called the rhizosheath. Not every crop, not every plant type does it, but grasses and corn does. The rhizosheath is where all the action occurs. You want that. Number one, you need root hairs, and it's biology that actually goes into the root tip that the plant strips nutrients from. A lot of those microbes die. Some of them go to the buds where the root hairs are. They secrete a hormone that induces the root hair formation. This is why we see so many more root hairs when we have diverse biology. And again, that rhizosheath is where nutrient exchange occurs, where free living microbes like Pivot Bio can do nitrogen fixation, and it's where water is retained and where the root is protected. So again, a free test. Pivot Bio, and I'm not going to say good or bad product. I'm a fan. I've got some friends that work there. Again, like I said, the energy problem is a challenge, but it's not diverse biology, so it can't trigger quorum sensing by itself. It needs other biology. Now, this is soybeans from Ankeny, Iowa. This is a third-party researcher. He's comparing us to a different microbial product that has a couple dozen bacteria and a yeast, a unicellular fungus. I'm not saying good or bad product. It can do some things like unlocking phosphorus and such, but this is their comparison. So mid-season, the grower's noticing with diverse biology, a lot more root mass, root hairs, better nodulation. This is another thing. When you've got that quorum sensing with diverse biology, the plant again changes its gene expression and will take in more rhizobia and form more nodules. So again, the complexity of the biology, it's just amazing how all these things work together. But what he's saying is his plants are basically 50% heavier here in the middle of the summer. And at the end of the year, he got about 6 bushels more an acre of beans. And the competitor product didn't work as well for him. So I could go on about that. Now I want to talk about residue. And this is kind of a pet peeve because, and I'll show you what I mean. But let's just look at, what you can leave on the field. And I know you've got a decision. Your corn residue, your crop residue has value. You can send it to feed, to make ethanol, whatever. But you also know you want to keep some of it out there because of the nutrients and the carbon. So if we look at corn, for every 40 bushels of yield, you have a ton of corn residue. So a 200 bushel an acre corn crop has 5 tons of residue if you leave it all out there. Each ton has 17 pounds of nitrogen, about 4 pounds of phosphorus, 34 pounds of potassium. Remember, potassium is a big part of the strength of corn stalks, soybean stems, etc., and some sulfur. All these, these are nutrients you've paid money on, you want to release. So how do we get that corn and wheat straw, etc., to break down? The only thing that will break down that residue and release the nutrients is microbial activity, period. It's not going to happen on its own. Microbes are required. So what factors influence microbial activity? Well, first of all, you need the right microbes out there. Second, it's the same as your crop. You need some temperature and you need some moisture. But, in a mat of residue, even morning dew can accumulate. So there's a lot of products out there, and I'm going to show you here in a minute. General rule of thumb, if the ground is frozen and it's winter, or you're so close to winter, you're not sure that you're going to get a couple weeks for the microbes to work, you probably don't want to spend your money on any product, including my own. You need a few weeks where you've got some temperature and moisture, because once it's frozen, or if it's just bone dry and there's no morning dew and there's no chance for water, again, you're not going to see much breakdown. So that's true across the board. So now let's go back to carbon and nitrogen ratio, and let's look at residue. So alfalfa, soy, etc., has more nitrogen, so more microbes can break it down faster. Your corn stover, though, for every nitrogen, 55 to 60 carbons. Wheat straw and other grasses, 1 for 80. They're very nitrogen poor. So you've probably seen where you can get tie-up of the nitrogen, especially if there's a lot of residue out there, and if you've only got a bacteria or a bacteria-dominant soil, because where are the bacteria going to get the nitrogen? It's not in the residue, it's in your ground. So they're going to steal it and tie it up. So what kind of products do you want? And I'm not going to name names, but this is a list of different microbial products. Most of them are only selling bacteria. Your soil, if it's tilled, is already bacteria dominant. There's not enough nitrogen for bacteria to break this stuff down. And bacteria don't secrete the right enzymes to break down cellulose and lignin. Those are the 2 biopolymers. They're 2 of the 3 most abundant on the planet. That's what makes your corn stalk strong. That's what makes a tree that's 100 feet tall stand up to a 90-mile-an-hour wind. They're tough. They're hard to break down. You need fungi and certain kinds of fungi. So our breakdown product, we, in our process, accelerate and promote the growth of those fungi really good at breaking down cellulose and lignin. And even fungi need some help because there's not enough nitrogen. So we actually put nitrogen in the product. The rest of these don't have the right biology, except product B, where they claim to have 70 bacteria and fungi. I can't find what they are, But if they've got the right kind of fungi, that could be a good product. That's the kind of thing I'd look for before I spent any money on a breakdown product. So this is our product. This is Iowa. 3 weeks before, so this picture was taken November 10th. The ground was vertically tilled. And then on part of the field, the diverse biology rich in fungi was applied. I want to stress this picture is a really good condition picture. This will not be everybody's experience because he had temperature and he had moisture. But look at the breakdown in his, basically saying, I'm going to sell my chisel plow. This helps me now. I can no-till because now I won't have this huge mat of residue that's going to have my ground heat unevenly and cause all kinds of problems when I try to plant through it. But this picture is a little deceptive. There's still residue out there to protect the soil during the winter, which we want. We don't want bare dirt. So again, the right biology can do the job for you. So with this, I'll just summarize. I gave you a lot, but I really hope you understand the science behind why, if you really want your soil to function, to get the most out of your nutrients, to get the most out of your crop. And again, I didn't even talk about how if you've got good beneficial biology, bacteria, actinomycetes, fungi, protozoa, amoeba, your soil will naturally suppress diseases. There's a whole body of science on that, what they call suppressive soils. Again, the benefits are incredible. You're going to get more using less while you actually build the quality of your soil, which makes your land more valuable. And I don't know if your plan is to pass it on to the next generation. You need tools that help you do that in a way that also help you stay in business. Biology can help. It's a tool. It's not a silver bullet, but it's a tool. So again, look for something that's got the diverse biology. And with that, Greg, I just really thank everyone for their attention, and I'd love to answer questions. I saw a lot pop up, but I didn't read any of them because that's the way I am. So
Right.
I get too focused.
Sure, you bet, Dave. Thanks so much. And yes, we are getting a lot questions from our audience. Just a reminder, you'll see that little speech balloon at the lower right-hand corner of your screen. Click on that, you can type into the box and then press the return key on your keyboard, and we'll get the questions to Dave. Dave, I want to start out just a couple things before we get to some of the questions from the audience. So it really sounds like this whole area of microbes, just to give us some context, is really in its, to me, it sounds like it's in its infancy. We've got so much that we don't know and we're still learning about. Help us frame that discussion on where we're at and what can we expect in the future in the whole thing with
So,
microbials.
it's when I started in this, and it was really the customers of Holganix, which are a lot of the professional golf, football and baseball stadiums, some of the top golf courses. So the best turf people in the industry convinced me I was wrong, that I needed to learn about soil health because they told me how much they cut their fertilizer, fungicides, watering. We are still so low on the learning curve because I would go and then meet different soil scientists and I'd get so excited. I've got so many questions. And I'd start asking them. Every time they'd go, ha, I've got the same questions. I'm like, oh, what do we know? So there's a lot we don't know. And I just really will be interested to see 3 years from now what I say that's different. So I don't ever want to mislead people, but we're on the learning curve and we're probably more toward the bottom than the top. But it's clear that the amount of science that's out there, biology like this, is a tool we need to really, really, really pay attention to. Down the road, someday, genetics companies are going to sell a suite of microbes with their seed because they're going to know those microbes are the ones tailored to the plant that they're going to spend energy and all these secondary metabolites to precisely attract those and repel others.
Mhm.
But we're nowhere near that today. So what I get from people that have been doing this a lot longer than me is go for diversity because the soil will figure it out. I hate that answer. I want to know, I want to be more precise. I want to make sure when people spend money, they're spending it on what they need and what they don't. But honestly, that's where we are.
Yeah. So kind of related to that, we have a question from the audience talking about, is there a published microbial population that represents a healthy functioning soil? Is there benchmarks out there?
Um, yes and no. I would say probably more no. We can get, it's more, it's not species. And I think this is really important with maybe a few exceptions. It's function. So we want microbial types that will unlock phosphorus. We want microbial types that will secrete hormones that induce rooting. We want microbial types that the plant root tip and rhizophagy will absorb to get the nutrients and induce root hair formation. And it's not any one species, it's a collection of different types. And so I'm part of the Soil Health Institute, and that's the direction that they're going as well. It's more what are the functionalities. I can tell you it won't just be bacteria, it won't just be fungi. Um, Elaine Ingham has a nice soil food web. I think that summarizes it. And I think what we find will fill in a lot of details through the years, but that soil food web, I think, will still pretty much be intact.
Mm-hmm. And related to that, Dave, is there some type of soil test or something like that that a farmer can use to identify and benchmark, his level of microbial populations?
Yeah, so we do things like Haney tests. There's the microbiometer test. They don't tell you at the species level, they'll tell you basically bacteria to fungal ratios and overall microbial activity. So doing that, we will see microbial activity. If you use our product at planting, stays higher throughout the season than where we didn't use it. But it doesn't get you into, it doesn't do protozoa, amoeba. You can do those genomic testings like through trace genomics and stuff, and they can give you a very detailed list, but it changes foot to foot, week to week. And candidly, when I look at that readout, unless there's things that are just glaringly missing, like, oh my gosh, there's no fungi, there's no amoeba, I don't know what to do with the data.
Mm-hmm.
So it's not something I generally encourage. I think look generally at your microbial activity with tests that are a lot cheaper. And like I said, in your corn or grass plants, if you have dirt sticking to the root, you have a lot of root hairs, that's free.
Absolutely. You mentioned, you briefly talked about, Holganix, your Bio 800 product. Can you tell us a little bit on how it's applied, when it's applied? Do you have flexibility on application, things of that sort?
Yeah, it's very flexible. So you can tank mix with anything. Remember though, anhydrous ammonia is really good at killing things. You don't want it splashed on you. So I'd rather you not lay our product right down on top of anhydrous. Don't tank mix with the fungicide, but you can go in furrow, 2x2. A burn down is good, or with the planter with the fertilizer or insecticide. But like I said earlier, don't wait long after emergence because we miss that really important burst of root development when there's germination. But I've got customers that do about everything under the sun. It's really flexible. That's about half a gallon an acre. Mixes easy, goes through whatever equipment, even drip.
Yeah. Does it require any special handling or storage?
So our distributors keep it refrigerated. Customers, especially if you get like a 250-gallon tote, it will be good. Just keep it out of direct sunlight anywhere 4 to 8 weeks. So you've got time to work through it. If you mix it in a tank on your tractor with fertilizer and you hit a long rain delay, this is living biology. It's going to grow. So if you're 5 days, 7 days or longer in that tank, it's not going to come out very easy. You're not going to be happy with me, but I can't help that. Again, it's going to plug 100 mesh screens if that happens.
Mm-hmm.
So
So this kind of leads up to a question we've gotten from the audience. This person asks, can you do a very light incorporation of the Bio 800 1 week before planting soybeans on corn stalks? Would this not be work better than spraying after planting?
I would like that, because what you've got is you've got food for the microbes, and the fungi are going to get to work breaking down that corn residue. And as long as we're using enough total liquid per acre, so I recommend 15 gallons, water, herbicide, whatever, not fungicide. I've had guys go as low as 10. So as long as we're wetting some into the soil, that works great. I've got a lot of people that do that. I've
Yeah.
got some people that will even split apply. Now I know when you're planting, you're trying to go hard, and maybe you don't even want to stop and put this in your tank, let alone do it twice. But some guys will put a quart at planting and then another quart when they side dress or top dress. It's flexible, but it
Mhm.
depends what you do. But then you might have to have separate deliveries, because now it's sitting out of refrigeration. Um, which is why I just say keep it simple, a half a gallon when you plant or close
Right.
to it.
Right. So another question from the audience, what's the lifespan of your product? Is it a yearly application required, and does latitude impact lifespan?
So lifespan in the soil is really hard to tell, because again, microbial populations change so much throughout the year depending on the weather, the moisture, and remember even what was planted the year before. So here's another thing. Corn on corn. In year one, the microbes in the plants accumulate what are called secondary metabolites, or different chemicals that stay in the soil. So if you plant corn again on that ground, it tells the corn plant you may have an insect problem, spend more energy making cellulose and lignin, which means you get less energy going into yield. Now that varies a little bit by corn genetics. So my point is, the soil is so dynamic. I I can tell you, generally speaking, microbial activity is higher, but what survives and what doesn't, I can't look anyone in the eye and say this does, this doesn't. I just know it changes. And winter absolutely will change it. Through time, as you build organic matter, and we've got some ground that we've applied this product for over 20 years, we cut the rate way, way back and only use it once in the spring.
Mhm.
And if you ever get to the point your soils are so good that you don't need it, God bless you. Good job. I see no difference, by the way, in latitude. I do say, though, if you've got a choice, put it on your roughest ground first. Slopes, hilltops, poorer ground, because that's probably where you have the least microbial activity.
Mhm.
And so adding it back will give you the bigger response.
Yeah. I mean, you mentioned that, there's so many factors that affect not only the microbial population, but their effectiveness. Does it ever reach a point where your soil reaches an optimum level so that, you can expect consistent results year after year? Is that possible?
I hope so, but I haven't seen science that proves it. But I would certainly hope so, and I guess it really depends upon where you are. So again, flooding is going to mess everything up. So there's no help for that. If you're next to a river that floods, or you got low spots, that probably will never happen on those spots. But in good soil, even where you get a hard winter, some microbes are more freeze tolerant than others. Now, maybe those are the only ones you need. Science hasn't caught up yet. So, but a product like this, I mean, you can brew your own compost tea. You can buy ours. It's not that expensive, whatever. But you can definitely notice the difference, and it doesn't take 20 years. You'll start seeing differences every year, especially if you're doing other things to help, like reducing your tilling. If you can do cover crops, great. Leaving some crop residue. All these things fit together.
Hmm. Yeah, you mentioned, the importance of, having an active, soil microbial population. We had a viewer ask, what about if you added extra carbon or nitrogen or sulfur as a feedstock? Would that be effective in relation to your microbial populations?
Carbon, depending upon the quality of your ground. So I've got some people that are, out west where it's drier, that will add extra carbon. We put a little humic and fulvic acid in our product. It's not much, candidly. It's enough to keep the micronutrients we add in a source, we're in a chemical state where the microbes can readily grab them. Extra carbon works, extra sugar is fine. You don't need to. There's a pound of molasses in every gallon. Um, I would not do extra nitrogen. Actually, this is, again, I go back to my previous point. If we're putting on fertilizer, especially N and P, way above removal rates, now we're discouraging the plant from attracting and feeding microbes. And so I've got guys that some will put down 300 units of nitrogen and spread a lot of manure, and I don't even want to sell them product anymore because their plants are not going to cooperate. So we need to back down. How much? Again, there's no formula, but I can tell you I have a lot of customers that were at 1 to 1.2 pounds or units of nitrogen per bushel of corn per acre that over 3 years of playing with the product are now down to 0.55 and 0.6. They're getting close to removal rates. Some guys on some soils, especially a sandy soil, may not be able to do that. You may have to be at 0.8 or 0.9, but I would, that's huge. If you want to get the most out of any microbial, you need it to work with the plant. Don't over fertilize it.
Mm-hmm. One of the comments you made during your presentation, which I thought was really interesting, you talked about how you see differences between corn hybrids and soybean varieties in their ability to farm microbes. And one of the questions from the audience, this person asks, do you see these biological tools respond better with conventional varieties versus the more improved genetic hybrids might be in reference to more transgenic, hybrids and varieties?
Yeah, I don't see a difference whatsoever in, just my background. I cloned my first plant gene in 1983. I was in the lab that developed, well, we didn't invent Roundup Ready. We found God did it in a glyphosate manufacturing waste stream in a bacteria. So we found what already happened. So I know the biotech and GMO extremely well. That's a big part of my career. I can't imagine any difference that would make it stand out versus conventional varieties. But every hybrid is going to have its own set of microbes in common with every other, and some that are unique, including GMO versus conventional. I can't tell you what they are though. I just know, and this is not my science, this is published science. I spend a lot of time reading what the universities are doing.
Right. And, as a company, how do you go about discovering or finding out which microbes do what, and, which ones are beneficials and which ones aren't? How do you go about that whole process?
To me, it's almost exclusively reading the scientific literature. And I don't do, the newspapers or anything. I go to the peer-reviewed scientific literature, things that most people don't want to read, and I understand why, because you need a cup of coffee to get through a lot of these things, or two, because they're dry, but they're rich in information. We do some of the research to confirm what we see with our products, but I depend on the scientific community. And fortunately, that's my training. That's what I've done for the last 40 plus years.
Mm-hmm, mm-hmm. And then we've got one more question here. Is there any negative interaction between glyphosate and biology or the biologicals?
So there are microbes that will make aromatic amino acids. So what does that mean in English? There's basically 20 common amino acids, or the links on the chain that make up proteins. Aromatic is a subset. That's what glyphosate inhibits. Some microbes make those too. So glyphosate would inhibit those. They would need to eat them like you and I do. We don't make them either. We have to get them in our diet. But glyphosate is quickly bound by calcium in the soil, and there are a lot of microbes that can degrade it. So what I haven't seen is what the impact is on different soil types, different glyphosate rates, and for how long. So glyphosate is not one I worry a lot about. I worry more about fungicides and really fumigants and putting down too much nitrogen and phosphorus.
Right, right. Well, David, it looks like we're at the end of our session today. First of all, I want to thank everyone who joined us today and those who asked questions. They were very great questions, and Dave, you did a wonderful job answering those and providing us with a wonderful insight on soil microbials and where it's all headed and the exciting opportunities and possibilities farmers have with biologicals, soil microbes to enhance their efficiencies and crop performance. So with that, thank you very much, everyone, and have a profitable day.
Yeah, thanks everyone. By the way, there's my email. So if there's questions you have, please contact me.
Great. Thank you so much, Dave, and thanks to
Thanks.
everyone in the audience.
Thank you.